Good afternoon, respected dignitaries, researcher. हाँ तो केला चालू. Good afternoon, respected dignitaries, researcher, and dear student. I am Dr. Swati Punkar. Welcome you all for this second session on behalf of Department of Botany, Shri Shivaji Science College, Amravati, which has jointly organized the 2 days national online workshop on fungal systematic and technological and advances in association with Mycological Society of India, Mumbai unit. It's my pleasure to mention that we are having Honorable Dr. Shilpa Verekar, Central Quality Assurance Laboratory, Parle Agro Private Limited, Mumbai, as a chairman for today's session. I kindly request her to chair the session. The speaker of this session is Honorable Dr. S.K. Singh, Scientist and Coordinator, National Facility, NFCCI, Agarkar Research Institute, Pune. Let me announce that this session will be followed by instruction for the assignment. Dear participants, kindly raise your queries in chat box during the session. Let me introduce in brief the chairman and the speaker of this session. Honorable Dr. Shilpa Verekar has completed her PhD in microbiology from Jivaji University, India. She is an industrial microbiologist with over 20 years of experience. She was senior research scientist at Research Center of Piramal Enterprises for 14 years. She was an assistant professor in Department of Microbiology at St. Xavier's Autonomous College, Mumbai for 2 years. She is actively engaged in the discovery of novel bioactive molecules, particularly from fungi, in the areas of anti-cancer and anti-infective diseases. She has 27 publications in reputed journal, 7 book chapters, and 1 patent to her credit. She was awarded the Women Scientist Award at International Conference on Mycology and Plant Pathology, Biotechnological Approaches at Banaras Hindu University in 2012. Currently, she is head of Central Quality Assurance Laboratory of Parle Agro Private Limited at Mumbai. It's our great pleasure to have such an eminent scientist of international repute as a speaker for this session. Dr. Sanjay K. Singh, who has graduated and post-graduated in botany from Kanpur University and doctorate in mycology from Gorakhpur University, Uttar Pradesh. He has allied postdoctorate fellowship, CSIR, New Delhi, Young Scientist Fellowship of DST, New Delhi, and Research Associated of Planning Commission proposed project at Goa University. In 2001, he joined MSCS Agarkar Research Institute, Department of Science and Technology at Pune as a scientist C. He is now Scientist F and in charge Biodiversity and Paleobiology Group. Dr. Singh was selected for DBT Associated Program for Young Scientists in niche area of agribiotechnology and visited Montana State University, USA, where he worked with world-renowned researcher Professor Gary Strobel and received specialized training in biology of fungal endophytes and their bioactive metabolites. Dr. Singh operated 14 projects and 8 extramural funded research projects funded by various government funding agencies. Dr. Singh was elected as a President, West Zone Indian Phytopathological Society, Council member of IPS and MSI. He is Fellow of Indian Phytopathology, New Delhi, and Honorary Fellow, Society of Biotechnology, India. Dr. Singh has published more than 100 research papers in international and national reputed journals and contributed 12 chapters in books. He has delivered more than 30 lectures at national and international events. Now, I request him to deliver his talk on the topic taxonomy of hypomycetes fungi.
Singh sir, please unmute your mic and switch on your video. Yeah. Please, sir.
Yeah, good afternoon. And at the outset, I would like to thank the organizer for giving me opportunity to share my thoughts about the subject, as well as I would appreciate the uh, the the kind of these webinars are being organized under the umbrella of the Mycological Society of India Mumbai unit. It's very exciting and inciting for the young researcher, MSc student, PhD student. And really I'm thankful for these organizer for such generous venture being organized, and it is really very very informative, and we feel that the mycology really the status of mycology need to be improved in our country. So without taking much time, I will move to my the presentation. Yeah. So since we are talking about the fungi, so we know that how fungi are critical, and at the same time they are really incredible as far as human life are concerned. These fungi have the ancient origins with the evidence, DNA evidence indicating that they first appeared around 1 billion years ago. Fungi are diverse in nature with around 144,000 species are classified and documented so far, which is at the rate of roughly 2,000 new species being described per year. This is the data from 2018. And from the Kew Botanic Garden, Professor Hawksworth and other colleagues, they came out with this report. The latest estimates suggest that the total number of fungal species on Earth is between 2.2 to 3.8 million. This number exceeds to estimated number of plants by more than 6 times. Means, in other way, the one plant is to 6 fungi. That is uh, the estimates, extrapolated estimates. Based on the extrapolation, and these figures has been given based on the extrapolations of plant and fungus species ratio, supplemented with the DNA studies of environmental sample. Though if you carefully look at the data, only 93% fungal species are unknown. 93% fungal species are unknown to the science. They incredibly touches every facet of life. Means only roughly 7% are known for us, for the science. But still, they are impart how they are critical and for our, as far as the human life is concerned. This is very interesting. The flow chart is given by the March in 1987. How these fungi are important for us. If you look at the upper arch, it tells the what are the uses of fungi, where these fungi can be used, whether it is biotransformation or extracting useful metabolite, enzyme, biological control, food yeast, mycoprotein, mycorrhizal fungi. If you look at the lower arch, these fungi also, they are really damaging also at this, like they can cause the animal and human mycosis, a huge loss due to the spoilage in the grains, food, mycotoxins. They are really allergic and they induce the allergic responses and plant diseases. So probably in the early stages, in early days, because of these problematic like a plant diseases, allergic response, mycotoxins, these fungi were not really considered as a good organism probably. So even some kind of scary, reflection still goes, and people probably are uh, do not like fungi much. But if you carefully look at the beneficial aspect, what not, fungi can give many things. Even in terms of economy, probably this upper arch will fetch more economy than the lower arch, which is known for the damaging economy. So as far as the fungal biotechnology aspects are concerned, really fungi one of the best system for the biotechnology innovations. So I'm not going much detail into that because of the time constraint. But yes, these are the area where the student and new researcher can look for these fungi. What are the beneficial aspects are there. We know that in if you talk about the forestry system, most of the means more than 80%, 90% our plants, they have the association with the mycorrhizal fungi. No mycorrhizal fungi, probably survival of the plant will not be there. And we know that what is the ecosystem services these fungi are offering for us. In nutshell, no fungi, no life. So now coming to the classification, just I will give the glimpse about the classification system. Earlier it is followed. The first classified by the Aristotle over 2000 years ago. The classification scheme of Middle Ages was replaced by the Carl Linnaeus, and he published Systema Nature in 1735. And we know that the Carl Linnaeus is known for the father of taxonomy, and his name is very famous with different name like Carl Linnaeus. Also is known as a Carl von Linnaeus, Carolus Linnaeus. So different texts you'll find in different books these names. One of the best and well-known the system given is the binomial nomenclature, which is still very much valid, and we follow that even today. If you look at the quickly classification system, the earlier the Linnean system, we know that it was a two kingdom system, and what was the key criteria followed at that time? It was the movement and responsiveness to external stimuli. Then Hague 1860 given the three kingdom system based on the division of liver, tissue system, morphological complexity, and mode of nutrition. Copeland, again 1956, gave the four kingdom system based on the nuclear organization, lifestyle, and biochemistry. Later way back in 1969, the fifth kingdom system was proposed by the Whitaker, you know, based on the mode of nutrition, lifestyle, polyphenism, unique biochemistry like chitin in cell wall. And then there the other system came like a two super kingdom system. So lot of advancement in the classification system of the organism has taken place in due course of time. So Linnean system, it was two kingdom system where only planta and animalia were described, and everything, all fungi were kept as a part of the plant. That too is a very simple, like a lower plant. It was considered as a lower plants, simple. So then the hierarchical classification system was developed, because while giving the different classification, the different researcher, they gave a different concept, and each and every classification system were based on certain concept. But there are many classification scheme. Each one can be argued to be more natural in one respect than the other. So it seems that as long as people will continue to make up such system, there will always be the disagreement also. Agreement and disagreement will always be there. So one point which has been agreed upon, that the closely related species are arranged into a genus, genera are arranged into the families, families into the order, order into classes, classes into the phyla, phyla into kingdom. So this arrangement, hierarchical arrangement, was definite, and most of the classification system given, people have followed this hierarchical system. So using this classification hierarchy, we can place any fungal species into the different category or taxa in various classification schemes. Now, some of the important uh, system here, I will just quickly explain that in the early days was highly qualified, followed by the people in the classification and characterization of the fungi, like Sacardo and groups. There's the classification based on the mode of grouping and pigmentation of these spores. You can see here that these two, hyphomycetes and coelomycetes, were characterized based on, these are the grouping system, and what are their basic character based on this arrangement, classification arrangement was given. Then the very interestingly, the based on types of spores, one classification, I would say these criteria, these concept, though it is obsolete now, but the system, the concept which was given by the earlier researcher, these still it is being followed. And in science, I believe that in this our mycological science, it will never be obsolete. And this terminology we must know, and especially for the beginner, for the student and PhD student, those have the people have the interest in the taxonomy and systematic, must know this system. Actually, it is not obsolete. We must know that basic fundamental. Look at here, the based on the morphology, and here they have given definition and type. Like here you can see the single cell spore is there. So in technical term, it is a unicellular, globular, or slightly elongated. So if it is a single cell, it is called as amerospore, amerospores, right? And there are two types, basically broadly it was categorized, like many people, you have seen that demataceous hypomycetes and non demataceous hypomycetes, means one group of fungi, they are like a hyaline, and the other group, they are darkly pigmented. So here the type, number of cells, and their color. Based on that, these classification was given by the researcher. So here we have, if it is a hylospore, then the other may be a pheospore, means darkly pigment. Like that, here we have the two cell system, bicellular. So we call it, technically it is called a didymospores. What is the type of the spore? It's a didymospores. Then we have more than two, so like a phlegmospores. Then we have like a muriform. There is not only the transverse septation, but the zigzag are like a transverse and longitudinal septa, both are there. Then it is kept under the dictyospores. Then unicellular or septate or very elongated like filamentous structure is there. Then it is called as scolicospores. Best example like Sarcospora, you know that you might have seen the spore. So the Sarcospora spores were kept under the scolicospores. Or sometimes if spore length is smaller, then it also comes under the phlegmospores. Here, the unicellular or septate, spiral or helicoid. So if it is a helicoid, then it is a like a helicospores. So very interestingly, based on the structure and color, this definition was given, and it is like in one look one can remember this. The another, this very interesting, is like a star-shaped spore. Sometimes we get a very unique group of fungi. So it is called as starospores. So this is very basic but very interesting. And sometimes when you sit and try to identify the fungi and record the characteristic feature, these type of terminology comes on the way, and you should know that. So as I said, that basically these fungi are presently it is splitted. The whole kingdom is splitted into three, where the fungi itself, the true fungi are there. Then stemini, phyla, like all Omycota, Omycetes fungi earlier, Pythium, Phytophthora, all those, they are separated. Then the other one is the protist, where the Plasmodiophorum mycota and other group, and even Myxomycetes, earlier it was part of the fungal kingdom, is separated. Important point, whether really the taxonomy is important. Why? Because always there is an argument between those who love the taxonomy and who do not like the taxonomy. But the fact remains fact. Here, very interesting quotes given by the Plotkin, the president of WWF, that as long as people think that genetic engineers can cure everything, the world will think that it does not need taxonomists anymore. But there is no biological diversity without the taxonomies to tell the stuff apart. Very true. The very first step is the crucial step. So taxonomy is not just morphology, but the summary of all that we know about the organism incorporated into the orderly system. So taxonomy knowledge and expertise, I would say it's really invaluable in designing the screening program also for the bioactive metabolites and industrial enzyme, especially when you go for filing the patent either for the process or the product. The first requirement is whether your organism dealing with whether properly identified and characterized or not. So there the taxonomies play very important role. But fact is that declining interest and expertise in classical taxonomy and classification of fungi is really, really a matter of great concern, not only in India, in globally. So I'm coming to fungi imperfecti because the title of my talk is about the hypomycetes. So how these hypomycetes are important and where to connect and how to go ahead, what will be the roadmap. So very often this term comes Many people, they use the term only single spores. So, and Conidia also you write, you get many books, monographs, and papers. So, what is this Conidia? Actually, it is asexual reproductive propagules produced by a large group of economically, medically, and ecologically important microbes, collectively referred to as conidial fungi. The industrial and these group deuteromycetes, imperfect fungi or hypomycetes, you can say that this is a very big group and very important, industrially important, products derived from many fungi belonging to this group like citric acid, gallic acid, gluconic acid, penicillin, griseofulmin, etc. Whatnot. It's really one of the like gem, I would say, this group. So important fungal parasites are plant, food spoilage, textile degrading, and most of those fungi causing mycosis are member of this group fungi. Majority of conidial fungi have no known sexual state. That is why even quite often you will get in the literature, this is called as a farm group or fungi imperfecti, and it was earlier categorized under the subdivision like Deuteromycotina. So what should be the criteria of identification? Considerable differences of opinion among the fungal taxonomy regarding the criteria for classification of this class, especially hyphomyces, is really, it was very important earlier, and lot of argument and researchers, their difficulty face the difficulty that what should be the valid criteria for this farm group of fungi. So conidial ontogeny, the concept was given, which has been of considerable importance. I will come later about the conidial ontogeny. And identification of hypomyces is primarily based on the microscopic morphology, where we see the major, characteristic feature like conidial morphology. What is the septation? What type of septation? What is the shape of conidia? What is the size, color, and what is the cell wall structure? Whether cell wall is the smooth or the rough structure, spiny structure, different degree of the roughness based on the those these criteria, the fungi earlier people used to identify. Arrangement of conidia as they are born on the chronogenous cells. For example, if they are solitary, arthrocatinate, blastocatinate, basocatinate, or gliosporic. So all this terminology I'll come later and I will explain that. So important is that these terminology one has to learn, and there are standard text available. What is the meaning when it is called as the here arthrocatinate means what is the meaning of the arthrocat? Unless you know the meaning, it is difficult to understand when you sit with the microscope and look in about your fungi. So type of chondrogenous cells like non-specialized or hypha like a phyllide, annelide, or sympodial. It may be any can be there in your fungus, and other additional features such as presence of sporodacia or cinnamata. It can be in a different group of fungi. And last and foremost, the sequencing and phylogenetic analysis. So, before the sequencing and based on the DNA data, these are the morphological identification were very, very important. And even in today, in the era of the genomics, these criteria of identification of fungi play a very important role. So here I'm coming to the hyphomycetes. There are the blastomycetes, coelomycetes. These are considered as the farm classes. So hyphomycete, how to first hand identify whether your fungus is belong to hyphomycete. In this case, the conidia from aggregated or separate chondrogenous cells bound on the exterior face of the substrate and not enclosed by additional fungal or host tissue. Like here, you can see the photographs here. Sometimes of the fruit body like structure is formed, and inside these structure there are coridia formations are there. So, but in case of hyphomycetes, you can see this, all these spores or conidia are formed directly on the coriophores from the conidiogenous cells. There is no envelopes or any coelom-like structure is formed here in this group of fungi. Quite often in literature, we get these terms, systematics, taxonomy, nomenclature, but they are very, very important and they are interlinked with each other. So how to, remember these things, that is also important, because for beginner it is quite confusing. These terms are often defined differently in different texts. Taxonomy originally means the principles underlying a system of classification, right? Presumably, a taxonomist was one who studied and applied these principles. The concept has broadened, an individual employed to study, identify, name, and classify fungi and act as the curator of the herbarium, right? So here is interesting just to remember this formula. So this will give the whole picture and concept. Formula expressing relationship of these terms may be as systematics is equal to taxonomy and nomenclature plus nomenclature. Here, when we sit for the identification of any particular fungus, then there are two type of analysis we have to do principally. If you carefully, if you look at it, is an analysis followed by synthesis. What we do, we collect the sample, prepare the slide, and analyze under the microscope. So what we are analyzing, we try to look for a different character which the particular fungus is showing. So it is like a analysis we are doing. Okay, whether it belong to the hypomycetes, it belong to the ascomycetes or what. Then we try to record the character, and based on those character, finally we come to the conclusion, okay, this fungus is belong to this particular group, this particular genus, and further we go to the species level. So first we do the analysis and then synthesis. Once the fungus is identified in a particular genus or a species, like we have synthesized and we have reached to our goal. And there we conclude that, okay, this fungus is like Cercospora personata, something like that. Okay. So what should be our roadmap for the identification? Here we collect the samples or specimens. Then we try to isolate the fungus from the sample we collect from the forest. Then incubate, we keep it for incubation and certain temperature based on our knowledge, based on the knowledge gained from the literature. And after certain time of the incubation at different temperature, then we try to observe the fungus. What is the growth? If growth is there, then whether sporulation is there? Because we try to search the characteristic feature based on which we can identify whether your fungus belong to this genus, this species, this class, or whatever. So, what are the characters we need to record? Is growth character, then texture, colony texture, whether fungus is producing any color, pigment, what is the margin of the colony, whether it is smooth margin or wavy or something different. Whether the pigment it is producing, it is soluble in the PDA agar, whether the fungus is producing any particular type of fruit bodies readily into the culture or not, or any type of other structure. Means we have to be really vigilant while making the observation after the incubations. Any type of a different structure, interesting structure, we should not leave it, and we must try to observe it. Then after the growth, making a slide is also an art. Sometimes in beginning, if you make the slide, you will get nothing, just a big rope like bunch of the mycelium, nothing else. So that comes with the experience. And many time, many I have seen in the laboratory, people are not using any stereo microscope. Simply they will open the plate and prepare the slide from any one place. So most of the times, these spores, when you touch the culture, spores are dispersed. So making a slide is also an art, and you should look the plate under the stereo and where the growth is there, what type is, if sporulation is there, carefully you have to touch with the fine needle of the tip of the needle, and then lift the material and put on the slide carefully. So unless your slide is clean, air bubble less, you will not get a very good picture. So it depends how you make the slides. In order to get a good picture, you have to be a very, your practice should be very unique and of the preparation of the slide. Then you can take the good picture, because nowadays very advanced microscopes are available. You can take a very good picture also. So these are the steps need to be followed. Basically, these lot of patience are required, but nowadays, I'm sorry to say that students don't have patience. Everybody want to just press the button and get the result. Anyway, it is not going to happen. So, in nutshell, best practices, intense learning ability, and practicing taxonomy is very important in decision making of the species level authentication. And it's not that the taxonomy is very dry subject. Beautiful color, beautiful texture you will get, from different fungi. If you isolate different types of fungi from different substrate, different habitats, really it's a world of amazing. You will really enjoy if you get very good, this how the colony texture look like. Here you can see this. Here the fungus is growing flat and very, you know, raise like structure, and also sometimes they produce the, something very hyaline or watery, exudates. That is nothing but something like a chemical substances. Here it's a actinomycetes. You can see the beauty of this actinomycetes, how they, simply on the water agar they produce, how it looks their colony. So even colony structure is very interesting in different fungi if you look. So now I'm coming to the slowly what is the conidium ontogeny? Why it is conidium ontogeny is important for the regarding the character from this farm group of fungi. So conidiogenesis means conidial development. There are two types. Here is like a blastic and thalix. Then again blastic into like a enteroblastic, holoblastic. Then under enteroblastic they are the phylidic, then analytic, then synchronous under the holoblastic, synchronous. All these, the type how it looks, I will come and tell you in the another slides. You can see here the conidium ontogeny is a very important steps to know that. What is that blastic? A conidium elongates and swell before being cut off by a septum. Usually originates at a narrow point on the condygenous cells. You can see here the diagram. Here, both the walls, they are taking place in the development. But here, when the upper, the tip region, they blow out, and after the certain times of maturity, they cut it here, and this structure is separated from the rest of the structure, which is called a spore, and they form the conidium. So that is the basic principle how the development takes place. Like that, sometimes both the walls, outer and inner walls, they take parts in the development of the conidium. Sometimes only inner wall take part into the development of the conidium. If it is inner wall only taking place, then in that case we call it um, enteroblastic. So there is a beautiful text available by Z.T. Cole in 1996 about the conidium ontogeny, and I would suggest those who have interest, we should read this literature, how the development are taking place. And sometimes while making a critical observation under the microscope, you will come to know all these things. So blastic is one and arthric is one. So here you can see this. It looks like this hypha. It is elongated. It is growing in different compartment, and each compartment or cell is separated, and it is forming a particular conidium. It is known as a spore. So, like if this kind of situation is there under the microscope, you can observe that it is means it is breaking with the septum. So it's come under the arthrospore, not the blastome. So like that, then many fungi, many group of fungi, the spore they produce from the phylotic condition. Best example is like aspergillus. I hope you remember the diagram of the aspergillus. Look at here. Here we can see this spores, means conidium. Then there is a macula where the spores are attached. Then vesicles and stipe. The whole part is a part of the conidiophore. But I can tell you the science has gone much and much ahead, and there are gold standards are set for these genera of Aspergillus, Penicillium, and fungi, and certain a very strict recommendation that what are the media need to be used. Your media should be what temperature, mean 25, 37 or different temperature. Then you have to record their characteristic feature, then different media and temperature. Then after that, so as we know that it's a time for the polyphagic approach. So after the morphology, we have to go to the genomics and then biochemical analysis. But as a beginner, we have to start like this. What is the stipe? Whether these stipes are smooth or rough walled, what is the color, then what is the vesicle shape, whether the conidiogenous region, what we call is the phyllite, like a conidium, what is the color of the conidium surface, how what is the spore surface is smooth or very porous, what are the, and then size, like maculae size, vesicle shape, size, all these character you have to record. Here, there are two basic mode of blastic development. As I told you that holoblastic and enteroblastic. Simply here you can see that in holoblastic, both the outer and inner wall, they are taking part into the development of the spore. But here, you can see that after some time, the only internal wall is coming out, blow out, and taking a part into the development of the conidium. Here, this is very complex, but it's a very interesting structure taken by the time lapse, microscope. This is a conical shaped tip of a fertile hypha slowly blows out to form the conidium initial over a 50 to 60 minutes period. So you can see this from here, this wall development has taken place, and here is started blowing, and to form a one particular, conidium to reach to its maturity, it took the 15 to 16 minutes. It was picture taken by the time-lapse microscope, and this is given in the book by G.T. Cole, 1996. And these are the very internal, very complex structure. What are the part, how it takes place. So if you have interest, you can go read it very carefully. You'll get a lot of information on that. So sometimes the repetitive formation of conidia from a single conogenous cell takes place. You might have heard the catenate, acropetal, basipetal, all this terminology. Unless you know the meaning of the terminology and how this development takes place, it is difficult to write a description of a fungus. So in case of catenate, means conidia forms in chain on one on the another, and still they remain intact in chain. But if it is non-catenate, if it is chain, the terminology uses catenate. If it is not in chain, after formation of the conidial development, then they get separated. So it's like a non-catenate here. And acropetal and basipetal, you know that acropetal, as the this diagram reflects that the first conidium initial is the bigger, then second is smaller, and third one is even still smaller. So this situation, if it is there in your fungus, then it is known as a acropetal. And basipetal is reverse. You can see this. The conidia on top is the bigger, then below it is smaller, and at the condygenous cell, it is still smaller. So acropetal and basipetal is one reverse from one another. Like that, phyllodic, many fungus, they are phyllodic, means it's nothing, they are coreogenous cells. They form a typical structure like a field. Field, you know that, like a colorate structure. But again, there in there are in colorate structure also sometimes color is very prominent. You can see under the microscope, sometimes you can't see that under the microscope. So these here in this diagram, you can see here the E, very prominent color. You can see that when the conidia are detached from the coreogenous cell, these type of colorate structure very prominently you can see under the microscope. So many fungi, they produce the they are the phyllodic, but many of them are non-phyllodic, blastic. We can say the blastic mode of development of the conidia. So here, phyllodic non-catenate conidia. The Phylophora verrucosa is one of the best example. You can see here very prominent phyllites. The conidia are produced from these phyllites, and after maturity, they get it separated. So again, the next conidia will come and produced, and they get released into nature. So these are like a, like a kumbh. Then analytic, the after the production of each and every conidia, their remnant, remnants remains there on the condygenous region. And condygenous region, they get increased in the length. And but there the something like a remnant remains there, and you can very well, you can observe under the microscope in the some fungi, where easily these analytic structure you will get in the chondrophore, in the chondrogenous region. Like best example is scopulary abscess brevicalis. You can see here these conidial chain are produced, but below there is a isthmus, like dark annelide structure. So annelide is nothing. Even if you look under the scanning microscope, you can count the number of these lines. That will give you that how many generations of conidia are produced from here. So very interesting, and sometimes in the simple light microscope also you can find this. So synchronous means sometimes conidia are produced directly. One conidia will produce, then again will come. But here there is a one condition like a synchronous conidiogenous cell, that conidia they will produce, then the conidiogenous region slightly shift, then again it will produce, then again shift, like this. This structure also you will get quite often in some fungi they produces, like cephalophora is one of the best examples. Exactly the fungus looks like that. When the spores they produce and the spore is still attached, you can see this like a bunch of the conidia on the conidiogenous region. Sympodium, one of the best example is the Sarcospora. Once conidia is produced, then it is shifted from one side. Again, second number conidia is produced. Then third number is produced. So zigzag like a sympodial condition, it will finally it appears on the corydiophore. Best example, Carvolaria. So one side, when it is produced, then slightly it is shifted, then again it will produce, again it will produce. So the when you look at the whole length of the conidiophores, you will come to know that okay, how which side conidia is produced, and you will get the final structure like a sympodial structure. Drexlera also, this comes under the same category. Uroconidia. It's very interesting. In case of the alternaria, very common, fungus, they produce a, pore-like structure. They leave behind a pore, scar-like structure. Like Sarcospora also, very prominent. So, blastic and thallic. Thallic, I told you that even these, if you know, the hypha breaks at the, septation, and that particular part turns into the spores, it's like a thallic development of conidia. And blastic, this situation was totally different. So holothallic, like holoblastic. So holothallic condition is also there here. You can see this. And best example in the microsporum, the fungus is a keratinophilic fungus. The holoarthric development with cyanolytic conidium liberation. I think move some slide here. Geotrichum, one of the best example, which comes under the category of holoarthric. Means it's a arthric. The spores they breaks into. Sometimes it appears that it is breaking at the septation, and they are formed, and every portion it forms as a part of as a spore. But when we call it holoarthric, means both the layer of wall they take part into development of the arthric development. But sometimes if uh, only inner wall or only one wall takes place, take part in the development, then it comes under the intero. So geotrichum is the best example of the holoarthric development of the convergences. Here is the enteroarthric development. You can see here this internal wall is taking place with the development, and from here you can see that it breaks, and every particular these cells is turned into the spore. Coccidioides Metis is the best example for this. Then endogenous mode of conidium. Quite often when we observe under the microscope, it readily appears that some spores are there coming directly from the uh, internal region of the conidigenous cell. Sometimes we don't come to know even. So best example is Ariobasidium pullulans. You can see this type of structure in the hypha, in the cell, under the between the septation, and they get released, they come out, and they are discharged in the nature. Sarcinic mode of conidium development means sarcinate type of. The best example is the Sarcinomyces species. Here you can see this. Very interesting. So this is overall picture of the mode of condogenous cell development. So yes, when the conidia they get mature and how they get detached from the uh, rest of the part from condogenous cell, that is also very important. And sometimes some genera it play a very important role as a unique character. So there are two types, like a schizolytic or rexolytic. So schizolytic are by fracture of the lateral wall between two septa. You can see this septation, fracture at the septation, but rexolytic, you can see here, here is septation, below the septation, from any place it can fracture, and this portion will get it separated and form the spore. Then successive development of conidial loci. See, one is like a, after one conidia will produce, then second will produce, then third. So there will be successive development, and it's like a progressive development. The coniogenous cell, coniogenous region or conidiophore length will increase. But there is another group of fungus which called as a retrogressive development, where the coniogenous cell shortens after each conidium is displayed, is produced. So this is overall a picture I have given about the development, and due to shortage of time, I think I will not go very much detail into that. So right from the leaf spot, when we collect the sample from the field, if somebody is working on the folliculus fungi, so what are the character you have to record? Leaf spot, type of leaf spot, distribution, then color, shape, margin, then fruiting structure, like production of conidia, whether the conidiophore are produced from the uh, stromata or only vegetative mycelium, whether secondary mycelium is there, what type of conidiophores, whether it is in fascicle, what is the color, shape, apical conidial scars, whether branching is there, then shape of conidia, color, septation, how is the apex of the, whether it is rounded, whether it is acute or something. So these are the character recording is very important, and that is what is once it is completed, means you have the characteristic feature of your own fungus. These are very, very important even in the era, today's era of the genomics. You can do the all PCR and phylogenetic tree, but the morphological characterization is also very, very important. It's not that the only molecular data complement to the morphology. Morphology also complement to the molecular data, that one should remember. Earlier, as I said, there are a number of different type of terminology for the shape also. In the dictionary of fungi, all these informations are available. One has to sit and learn that if it is spherical, whether globose, subglobose, ellipsoidal, all shape of conidia, these are very important feature uh, as an important part of the recording different structure. So, as I said, that modernization of mycology, the 20th century has seen a modernization of mycology due to advances in biochemistry, genetics, molecular biology, and biotechnology. Use of DNA sequencing technology and phylogenetic analysis has provided new insight into the fungal relationship and biodiversity. Challenged traditional morphology based grouping in fungal taxonomy, actually. The latest classification which came, it's a seven phyla system by the Hibbert and al. We know that. See, the earlier classification system was based on the morphology and physiological character, more on that. But later, in the new classification, DNA-based classification has come, and a lot of changes had taken place even further to this. See here, these evolutionary studies says that fungi are more closely related to animals than any other kingdom of life. Here it is given 8 fungal phyla along with estimates of the number of described. This estimate was given in 2008. What are these 8 phyla? Like here, you can see this Cryptomycota, Microsporidia. Cryptomycota, this having around 30 species. Microsporidia, 1,250 species. Then we have Blastocladiomycota, Catridiomycota, Zupecomycota, Micromycota. And then we have the Ascomycota and Basidiomycota. So at the base, you can see here that it is basically divided into eukaryotes and prokaryote. Prokaryote, we have the like archaea and bacteria. But under the eukaryote, as I said that the fungi are more close to animal than the plant and other kingdom. So this is a schematic representation is given in the state of the worldfungi.org, and this is record of 2018. Then I'm not going into detail about the molecular identification, how to go ahead. The simple step we have to follow, you have the pure culture, extra pure culture, then DNA isolation, PCR amplification, then you have to uh, do the amplicon run on the gel, then chromatogram you have to generate, blast analysis, then multiple sequence analysis, and then phylogenetic tree constructed. I know the students are more interested on these things, but the earlier things is also very important. The who will bring the sample for you from the forest, and who will do the culturing and basic identification, conventional identification. So if one want to be a perfect in this area, then you have to go for the conventional as well as molecular taxonomy statistics. So here, without taking much time, here is the what we are doing at our center. So overall, in nutshell, the uh, our department, we have more than 1,200 publications from our department, and 88 people have taken their PhD and MSc degree from this department. We have 7 monographs. We have internationally recognized repository at ARI. National level workshop we have organized, and about 200 people, even faculty, more than 200 people, faculty, PhD student, MSc student, and people from the industry has taken, um, training at our center. We have organized 5 international and national conference at this School of Mycology. Many extramural funded projects has been operated at this center. So the services portion which we are offering, it has gone to more than 700 different organizations across India from 25 states and 5 union territory of India. And presently we have more than 5,000 indigenous fungi conserved at our centers. So in nutshell, we are serving for the nation at this DST National Facility at ARI. It's really because of these two giant personalities who really laid down the foundation stone here at this center, about the very well known school of mycology where we are sitting, and really we are uh, very much grateful to these uh, founder member of our department and our also as our institute. So we pay our tribute to these founder members. Because of them, we are here. Many people might have attended the recent international, symposium which we had organized in Pune. So this is some memory from that. Thank you so much. Thank you.
Thank you, sir. It was a very informative talk. We are highly thankful to you, sir, for addressing us on such an interesting and very informative subject. Now, dear participants, the topic is open for discussion. Here is one question from Manohar Gavit, Satna College, Nashik. When we collect the fungal species from forest, so is there necessary government letter for permission collection of specimen?
Yes, very much essential now, because these rules and regulations changes quite fast. So now I would advise that before going for the forest area, if anybody is working in college or university, better to intimate to the concerned biodiversity authority. And if you there is a simple form available, you have to fill up, you have to send that form, and they will give the approval. So it may take a month time to get the approval, but yes, at least you have to intimate to the biodiversity authority.
Next question from Nadia Ashraf. Sir, how can we make clear drawing of fungal structure?
Yes, if you really want to, learn the beauty of, drawing, then you have to go for the basic system earlier, camera listed drawing. I am not sure that how many laboratory having the very conventional camera listed drawing. But if anybody is having in the lab camera listed drawing, you must practice it. Really, you will enjoy. As I said in my talk, that first analysis, then synthesis. You have to, draw the different picture, and obviously different picture. When you make the slide and put under the microscope, many structure they, scattered. They are not intact. Then you have to you separately draw the pictures, and using your skill and basic principle, especially the conidium ontogeny, if you know that, then you have to really visualize that, okay, this particular spores were attached here, or one on the other, whether it is in chain, whether it is in without chain. All those things you will learn that while learning the camera start drawing. Otherwise,
The
many
next question is there any protocol or method for preservation of fungal specimen which helps to preservation of herbarium? The question by Manohar Gavit.
yes, there are very good handbook of mycology, very old by the Hawksworth, and if you have that book, and I think it's a 1976 year of publication, but maybe outprint it is. But many library where the mycological schools were there earlier in India, they must be having that. So it's a very simple procedure. You bring the collection from the forest, and very next day you have to really press it. If you don't have press properly, then you can use the newspaper also. Means certain modification you can do that, because pressing is very very important. If specimens is properly pressed and dried, then it will remain for years together. Even sometimes many bigger herbaria, they have a 100 years old specimens properly locked in the herbarium. So that is one of the best method, and one should learn that how to make the herbarium. Like how the people working on the plant taxonomy, they are also preparing the herbarium on the herbarium sheet. The more or less the system is similar, but here in the mycological herbarium, what we want to preserve more on our fungal part. If it is a symptom on the leaf, properly leaf, because different type of plant having a different types of leaf. What maybe like if you take the mango leaf, it's very hard, so you need a more pressure to press it. On the other side, if you have a coriander leaf, it's a very fragile leaf. If you put the coriander leaf between the mango and any other harder leaf, then probably the chances of getting spoiled this will be more. So slightly you have to use the common sense also, but yes, technique one should learn that, how to prepare the herbarium.
Sir, last question by Parikshana Mathur. Sir, which class of fungi is generally known to be endophytes?
Many, uh, mostly they belong to the hypomycete, I mean ascomycete also. But I think there are some earlier report also, this basidiomycete also means sterile group. It's non-conidial fungi also. Sterile means unless you make some effort to declare that your fungus is sterile, it is difficult to say that it is sterile, because there are different classical method to induce this sporulation. If really fungus belong to the hypomycetes or ascomycetes group, and if you make an effort by changing either the media, by changing the physiological condition, or adopting a particular method, like grass leaf method is established by Professor M.C. Srinivasan. That was initially for the seed-borne fungi, but it works in many of the hypomycetes endophyte also. We have practice here, experience here in our laboratory.
Thank you, sir. Thank you so much. Now it's time to listen for remark of Honorable Chairman. So I request Dr. Shilpa Verekar, Madam, to kindly express her remarks.
Good afternoon, everyone. Am I audible?
Yes.
Yeah. So thank you, Dr. Singh, for giving a very interesting talk on a taxonomy of hypomycetes fungi. In his talk, Dr. Singh mentioned that fungi were discovered 1 billion years ago. There are about 2.2 to 3.38 million fungi which are existing, but only 7% are known to science. So 93% are unknown to us. He highlighted the importance of fungi as friends and foes. The classification of fungi was explained based on spores. He rightly said that taxonomy is not just morphology, but summary of all what we know about the organism. Dr. Singh spoke on fungi imperfecti and its industrial importance. He described the criteria for identification of hypomycetes by using classical and molecular methods. He emphasized that along with molecule, one needs to also focus on the classical techniques. He explained the roadmap of identification and conidiogenesis, giving suitable examples like Drusillera, Scopuloropsis, Orobasidium, and many more. Well said, Dr. Singh, that taxonomy is not a dry subject. That it is quite colorful. People always take it, kid, what am I going to do? Sit under the microscope and observing the plate. But believe me, it is very, very important because it is the identity of a culture, especially when you are working with industrial cultures or you're, dealing with the patent protection or the IP rights. Yeah. So I should only conclude that what Shakespeare has once said, that what is there in a name, there is too much there in a name. Thank you.
Thank you. Thank you so much.
Thank you. Thank you so much, madam. Before concluding this session, and proposing vote of thanks, dear participant, stay with us for important announcement regarding the assignment. So over to you, Mr. Avinash Darsimbe, for further announcement.
A very good afternoon to all of you. I, Avinash Darsimbe, welcome you all to the assignment segment of two-day national online workshop on fungal systematics and technological advances. So first, I like to share the guidelines of assignment and assessment competition. The competition will comprise of assignment and quizzes. It will be a unique competition to explore the world of mycology. There is an assignment for day 1, and you have to submit it till 10 a.m. of 13th November 2020, means till tomorrow. The quiz of day 2 will be conducted immediately after the technical session 1. The participants will have to complete it in half an hour. Both these activities will be conducted on the Google form, and their links will be shared on Telegram group, WhatsApp group, YouTube, and the website of our college. The top 3 winners will be awarded by merit certificate, each from the research, PG, and UG category in valedictory function. Both assignment of day 1 and quiz competition of day 2 will be considered to finalize the winners. The link of today's assignment, that is assignment of day 1, has been posted on Telegram group, WhatsApp group of online workshop, YouTube, and on college website also. All the instructions regarding the assignment has been mentioned in the assignment Google form itself. Please read all the instructions carefully. Please accept our best wishes for this competition. We are eagerly waiting for your huge responses. I also take an opportunity to propose vote of thanks. On behalf of Shri Shivaji Science College, Amravati, and Department of Botany, first of all, I extend my most sincere thanks to Honorable Chairman Dr. Shilpa Warekar from Parle Agro Private Limited, Mumbai, who has spread her valuable time from busy schedule to grace the workshop. I would like to especially thank our today's speaker, Honorable Dr. S.K. Singh, Senior Scientist, Agarkar Research Institute, Pune, for sharing his crucial work. I am also thankful to all the participants for their active participation. With this, we conclude for the day 1. Kindly join at 11 a.m. tomorrow for the next session, wherein Honorable Professor N.S. Atri sir will guide us. Thank you and stay tuned.